My name is Gülnarə Vahabova. Welcome to my lecture, and today we talk about hormones. The main function of endocrine glands is to secrete hormones directly into the bloodstream. Hormones are chemical substances that affect the activity of another part of the body. It is called target site. Hormones synthesis is a very low concentration and can have very powerful effects. And in our body, synthesis many hormones, and most of these only affect a few types of cells. The specific cells which are affected by a hormone are called target cells. Hormones influence the target cell by binding to proteins or its nature of membrane receptors, glycoproteins. Only the target cells for a certain hormone have receptors that we recognize and bind to that hormone. Hormones serve as messenger controlling and coordinating activities throughout the body. Also, hormones controls the function to entire organs, affecting such diverse processes as growth and development, reproduction, and sexual characteristics. Also, hormones influence the way the body uses and stores energy and control the volume of fluid and the level of salts, sugar, glucose in blood. Very small amount of hormone can trigger very large response in the body. Also, hormones circulate throughout the body. Each type of hormone influences only certain organ and tissue. In this diagram, you see endocrine system, and the endocrine system consists of a group of glands and organs that regulate and control various body function by producing and secreting hormones. It is main endocrine gland, and you see it's hypothalamus and pituitary gland, the brain, thyroid gland, parathyroid glands. It's regulate level of calcium in the blood. Pancreas regulated level of sugar in the blood, generally function, and synthesis two type of hormone, main hormone, insulin and glucagon. Adrenal glands secreted two type of hormones. It is catecholamines. Main hormone is adrenaline, catecholamines, and cortex secreted steroid hormones, gluca and mineralocorticosteroids. And sex hormone secreted in ovary and testes, female and male. So Now, in this, we consider classification of hormones, and in this table, you see uh, chemical classification of hormones. And as you see in this from this table, we divided hormones for 4 groups. First group of hormones, it is peptide protein or peptide hormones. Its name, it's hypothalamic hormones. Pituitary hormones have peptide nature, insulin, calcitonin, parathyroid hormones, glucagon, and other. These hormones consist of amino acids, as all proteins, and these hormones are water soluble, circulated in the blood predominantly, and peptide hormones are enabled to cross the cell membranes of the target tissue, and they bind to receptor of this membrane. These hormones cannot be administered orally because they wouldn't be digested in the gastrointestinal tract. And instead, they are usually administered by injection, for example, insulin. The next group, it is steroid hormones, are produced by adrenal gland, cortex, testes, ovaries, and pancreas. They are lipid soluble, synthesized from cholesterol or steroid hormones, derivatives of cholesterol. These hormones cross cell membranes readily and bind to receptors found intracellularly. However, because their lipid solubility renders them insoluble in blood, steroid hormones are transported in the blood bound to protein. They could diffuse out of the cell, and physiological regulation of their release wouldn't be possible. Steroid hormones are absorbed easily by the gastrointestinal tract and may be administered orally. Next group, it is derivatives of amino acids, but amino acid derivatives. It is catecholamines and thyroid hormones. The hormones are derivatives of amino acid tyrosine and hormone melatonin synthesis from amino acid tryptophan. Thyroid hormones mainly insoluble in the blood and are transported predominantly bound to proteins. But catecholamines are similar to proteins hormones, and these hormones soluble in blood are transported in an unbound form. And next class, it is special class. It's we depend it compounds to hormones. It is prostaglandins, thromboxans, and prostacyclins. They derivatives of fatty acids. So we consider the regulation of secretion hormones, all endocrine control. Main endocrine glands are controlled by the interplay of hormonal signal between the hypothalamus located in the brain and the pituitary gland, which site which sits at the base of the brain. This regulation is called the hypothalamic pituitary way. The hypothalamus, it is a small region of the brain that connects to the pituitary glands. The hypothalamus secreted several hormones that control the pituitary gland. Hypothalamus, synthesis specific hormones, releasing hormones, and these hormones divided for two groups, liberins and statins. Liberins stimulated pituitary secretions hormones, but statin inhibit secretion of pituitary hormones. The pituitary gland, sometimes called the master glands, because it secreted hormones that control the function of many other endocrine glands. In pituitary gland synthesis tropic hormones, all hormones it's called trop, for example, thyrotrope, adrenocorticotrope, or lipotrope hormones. These hormones synthesis and transporting to the endocrine gland, and in the endocrine gland by the action of this enzyme synthesis specific hormones. For example, thyrotrope hormone stimulated thyroxine, and this hormone secreted to the blood. It's illustrated in this picture. In the endocrine cells, synthesis specific hormones which secrete to the bloodstream and transporting to the target cells. Some hormones affect only one or two organs, whereas others have influence throughout the body. For example, thyroid stimulating hormone, it's hormone of hypothalamus, is produced in hypophysis, produced in the pituitary gland, affect only the thyroid gland. In contrast, thyroid hormone produced in the pituitary affect only the thyroid glands. In contrast, thyroid hormone produced in the thyroid gland affect the cell throughout the body and involve the such important function as regulation growth of cells, controlling the heart rate and affecting the speed at which calories are burned. Also insulin, other example, secreted by cells of the pancreas and affect the metabolism of glucose, protein, and fat throughout all body. Hormones after condition inside the cell are activated at specific times. So they must be turned on and then turned back off. Most hormones are regulated by feedback mechanism. Let's show the feedback mechanism. A feedback mechanism is a loop in which product feedback to control its own production. Most hormone feedback mechanisms involve negative feedbacks loops. Negative feedback occur when a product feedback to decrease its own production. For example, the thyroid hormones is a good example of this type of regulation. The hypothalamus secreted releasing hormone synthesis in hypothalamus, for example, which stimulated the pituitary gland and to produce TSH in pituitary gland synthesis thyroid stimulating hormone of TSH. This hormone interstimulated the thyroid gland, endocrine thyroid gland, to secrete its hormone or thyroxine. When the level of thyroid hormone is high enough, the hormones feed back to stop the hypothalamus hormones, or hypothalamus hormone from secreting a thyrotrope releasing hormone and the pituitary from the secret TSH. And this is a long lobe negative feedback, it is short lobe negative feedback. Without the stimulation of TSH, the thyroid gland stops secreting its hormones. Soon, the level of thyroid hormone starts to fall too low. In this picture, you see mechanism of hormonal action. And we have in our body different mechanism of hormonal action. It depends of nature of hormone and location of its receptor. In this picture, you see mechanism of hormonal action for peptide hormones, protein, peptide, amino acid, except thyroid hormones. Hormones activate the target cell by diffusing through the plasma membrane of the target cell. It's depend for soluble hormones to bind a receptor protein within the cytoplasm of the cell, or binding a specific receptor protein in the cell membrane of the target cell. It is for water-soluble proteins, water-soluble hormones. Specific receptor of this hormone are protein molecules. There are extracellular locate and present in the cell membranes. It is receptor of hormones. This hormones is first messengers, and because they cannot enter the target cell, produce second messenger with the help of the receptor that regulates cell metabolism. The second messenger may be different. It's, for example, CMA, cycle adenosine monophosphate. It's maybe calcium inositol triphosphate, but it's depend of hormones, and each hormone has specific secondary messenger. Present in the cell membrane are the receptors, G proteins, and adenylate cyclase. It is G proteins, what is a membrane receptor, and G protein. G protein are associated with GDP, diphosphate, and after this guanosine diphosphate, the hormone connect with receptor protein, causing a change in shape due to which it comes in contact with G protein. It interacts with G protein, change GTP or GDP to GTP, and after this The G protein activated with GTP adenylate class, with adenylate classes, and adenylate class convert ATP to cyclic adenosine monophosphate. The cyclic adenosine monophosphate activates the kinase enzyme, which triggers intercellular biochemical change like enzyme activation, secretion, and other. These biochemical change result in physiological and development effect. In the next picture, this picture, you show the mechanism of action for steroid and thyroid hormones. The steroid hormones. And these steroid hormones we say about before, it's next class of hormones, are derived from cholesterol, and they are not soluble in blood and can pass through the lipid barrier of the cell easily. In order to move through the body, they are encased with the water-soluble proteins that can travel through the blood. When they reach the target cell, they are released from the protein carrier and can pass into the cell and bind to internal receptor. The receptor of this steroid hormones receptor will be located in the cytoplasm. They have intracellular location. They bind to internal receptor, and this new hormone receptor complex, so this is hormone receptor complex, will cause the production of messenger RNA and codes for protein production, causing the responses of the cell. This steroid hormone stimulated synthesis of new proteins. It is main difference between action. In this picture, you see main hormone of hypothalamus and pituitary gland. The hypothalamus pituitary complex is a command center of the endocrine system. This complex secreted several hormones that directly produce responses in the target tissue, as well as hormones that regulate the synthesis and secretion of hormones of other glands. This is hormones of hypothalamus and pituitary gland, and hormones of hypothalamus called releasing hormones, which stimulated synthesis of secretion of pituitary gland. The main of hormone, growth hormone. These hormones, it's promote growth of body. Prolactin, also prolactin, promotes milk production from mammary glands. Thyroid stimulating hormone TSH, it's have different nature. It's glycoprotein, have carbohydrate in this compound and stimulates thyroid hormone release from thyroid. Next, adrenocorticotrope hormone. What's adrenocorticotrope hormone? ACTH stimulates hormone released by adrenal gland cortex. Follicle stimulating hormone FSH. It's also glycoproteins, and it stimulates gamete production in gonads. Also, luteinizing hormone. It's also glycoprotein. Its nature is also glycoprotein. It stimulates androgen production by gonads. Melanocyte stimulated hormone. Melanocyte stimulate hormone. It stimulates melanin formation in melanocytes. Also, oxytocin and vasopressin. As you see, the synthesis in hypothalamus, but stored in pituitary gland. Oxytocin stimulates uterine construction during childbirth, but antidiuretic hormone, it's vasopressin, stimulates water reabsorption by kidney. And now next we describe main endocrine glands and begin from thyroid glands. The main thyroid glands, and the first step, the hypothalamus secreted thyrotropic releasing hormone, which stimulated secretion in pituitary gland, TSH. TSH stimulated synthesis in thyroid glands. Three types of hormones: thyroxine, T4, T3. In general, we say monoiode, diode, and iode we say generally. And other hormone is calcitonin. Calcitonin regulated calcium, amount of calcium in blood. Main function of thyroid glands is to make hormones T3 and T4, which are essential for the regulation of metabolic processes without body. The hormones are constructed by adding iodine, results to the amino acid tyrosine, which is a major compound of protein, thyroglobulin. Thyroglobulin is produced in the endoplasmatic reticulum of the thyroid follicle cells and excreted in the colloid follicles. Within the follicles, iodinization of it is adding of iodine to tyrosine occur. The compound that is formed, first step, it is monoiode tyrosine when connect only one iode, monoiode, which is a single iodine added to the tyrosine. Through the action of the enzyme 3-iode peroxidase, formed second iodine added to monoiode tyrosine, and by the action also of this enzyme 3-thyroperoxidase, and form diiode tyrosine. It is diiode tyrosine. When two molecules of diiode tyrosine combine, and by action also of this enzyme thioperoxidase, formed tetraiode tyrosine. Tetraiode tyrosine, or other name, is thyroxine. It is main hormone of thyroid gland. From thyroxine, synthesis form triiode tyrosine. Thyroxine, tetraiode thyroxine, lost one molecule of iode and form 3T3, or 3-iode tyrosine. It is two main hormones of thyroid glands. It is a process catalyzed enzyme deiodinase, which require the cofactor selenium. Upon release of the thyroid hormones into circulation, thyroglobulin is largely conserved and recycled with the thyroid follicles. If we say about mechanism of action hormone, its mechanism of action like steroid hormones. And thyroid hormone interact with receptor to change genomic activity and affect the synthesis of specific protein during development. Thyroid hormone change brain function in adult life in ways that the both resemble and differ from their action during development. Receptors for thyroid hormones are intracellular DNA-binding protein that function as hormone-responsive transcription factors, very conceptually to the receptor for the steroid hormones. Thyroid hormones enter cell through the membrane transporting proteins. A number of plasma membrane transporters have been identified, some of which require ATP hydrolysis. The relative importance of different carrier systems is not yet clear and may differ among tissue. Once inside the nucleus, the hormone binds its receptor, and the hormone receptor complex interact in the specific sequence of DNA in the promoters of a responsive gene. The effect of the hormone receptor complex binding to DNA is to modulate gene expression either by stimulating or inhibiting transcription of specific genes, at the end synthesis of new proteins. If we say about biochemical effects of thyroid hormones, we talk about metabolism of proteins, carbohydrates, how thyroid hormones regulated protein, carbohydrate, and lipid metabolism. Thyroid hormones stimulate different metabolic ways, most tissue, and increase the metabolic rate. Increase body heat production, which seems to result at least in part from increased oxygen consumptions and rates of ATP hydrolysis. Also, its specific include increased thyroid hormones, increased levels stimulate fat mobilization, and increased consumption of fatty acid in plasma. They also stimulated oxidation of fatty acids in many tissues. And finally, plasma concentration of cholesterol and triacylglyceride are inversely correlated with thyroid hormone levels. It is one of the main markers of hypothyroidism is increased blood cholesterol concentration. If we say about carbohydrate metabolism, thyroid hormones stimulate also all aspects of carbohydrate metabolism, including increasing of insulin and depend enter glucose into cells, and increased gluconeogenesis stimulates synthesis of glucose, and also glycogenolysis, degradation of glycogen, and generate glucose, free glucose. Thyroid hormones also increased heat rate. And if we say about proteins metabolism, as you see, they act by two ways: and increased protein synthesis and degradation. It depends of organ and tissue. That's why normal level of thyroid hormone are essential to the development of the fetal and neonatal brain. Now we talk about thyroid disease states. We have two types: increased production of hormone and decrease. And disease is associated with both inadequate production and overproduction of thyroid hormones. Hypothyroidism is the result from any condition that result in thyroid hormone deficiency. Also, maybe iodine deficiency. Iodine is absolutely necessary for production hormones. Without iodines, hormones cannot be synthesized. Primary thyroid disease, inflammatory disease of the thyroid that destroy part of the gland, are clearly an important cause of hypothyroidism. Common symptoms of hypothyroidism arising after early childhood include weakness, hair loss, and reproductive function. And you see in this picture, the clinical condition is called myxedem. In the case of iodine deficiency, the thyroid becomes inordinately large and is called goiter. Hypothyroidism is a young children with congenital thyroid deficiency called cretinism. If that condition is not corrected by supplemental therapy soon after birth, form irreversible growth and mental retardation. Other type, hyperthyroidism, it is overactive thyroid glands occur when thyroid gland produce too much of the hormone thyroxine. Hyperthyroidism can accelerate body's metabolism, causing unintentional weight loss and rapid or irregular heartbeat. And you see in this picture symptom, eyes, goiter, and maybe also increased head contraction. It's called tachycardia. Used in this disease, used antithyroid medications and radioactive iodine to slow the production of thyroid hormones. Sometimes hyperthyroidism treatment involves surgery to remove all or part of a thyroid gland. Next, hormones of pancreas, and now we described hormones of pancreas gland. The pancreas is unique is that it's both an endocrine and exocrine gland. The pancreas had the dual function of secreting hormones into blood, it's endocrine, and secreted enzyme through duct, it's exocrine function. The pancreas belongs to the endocrine and digestive system, with most of the cells working of the digestive side. It's more than 90%. Now in this picture you see hormones which synthesis in the pancreas, The production of pancreatic hormones including insulin, somatostatin, gastrin, glucagon, and these hormones play an important role in the maintaining sugar and salt balance in our body. Main hormone of pancreas is insulin. This hormone regulates blood glucose by allowing many of your body's cells and absorb and use glucose. Insulin and glucagon cells are hormones which secretes by cell of pancreas. Beta cells secrete insulin, alpha cells secrete glucagon. Also, pancreas synthesis somatostatin. When levels of other pancreatic hormones such as insulin and glucagon get too high, somatostatin is secreted to maintain a balance of glucose. Also, other polypeptide of unknown function, or maybe this hormone helps control water secretion and absorption from intestinal by stimulating the intestinal cells to release water and salts into the intestine. Also here you see insulin and amylin. Amylin is a peptide hormone that is co-secreted with insulin from the pancreatic beta cell and is thus deficient in diabetic people. Also, it inhibits glucagon secretion. Insulin is synthesized in significant quantities only in beta cells in the pancreas. The insulin is translated as a signal chain. Pressures called preproinsulin. It is first step, preproinsulin. And removal of its signal peptide during insertion into the endoplasmatic reticulum generates proinsulin. It's first from preproinsulin formed proinsulin. With removal of this signal sequence formed proinsulin. And proinsulin consists of three domains: an amino terminal and terminal part, here a beta chain. From C-terminal part, carboxy terminal, it is A chain, and connecting peptide in the middle, known as the S peptide. This is C peptide, and within the endoplasmatic reticulum, proinsulin is exposed to several specific endopeptidases which excite the C-peptide therapy, generating the mature form of insulin. The mature form is main hormone. It's insulin, consists of two chains, A and B, which connecting by a disulfide bond. When the beta cell stimulated insulin is secreted from the cell by exocytosis and diffuses into islet capillary blood. C-peptide is also secreted into blood but has no biological activity. In most species, the A chain consists of 21 amino acids, and the B chain here consists of 30 amino acids. Insulin is secreted in primary in response to elevated blood concentration of glucose. Also, there is always a low level of insulin secreted by the pancreas. The amount secreted into the blood increases as the blood glucose rises. Similarly, as blood glucose falls, the amount of insulin secreted by the pancreatic islets go down. Insulin has an effect on a number of cells, including muscle, red blood cells, and fat cells. These cells absorb glucose out of the blood, having the net effect of lowering the high blood glucose level into the normal range. In this picture, you see effect of insulin to all metabolism. Insulin can be considered the anabolic or building hormone. It's show it in this picture at stimulated protein synthesis. It assists processes that build compounds for storage and decrease processes which break down those storage reserves. And its anabolic effect, it means protein, for example, protein synthesis, stimulated amino acid transport in the cell and stimulate protein synthesis. Also stimulate lipogenase, synthesis of lipids in adipose tissue and liver. And in muscle and liver, it is depend to carbohydrate metabolism. In muscle and liver, insulin increase glycogenesis and decrease glycogenolysis, or it's stimulated synthesis of glycogen. And that's why insulin decreased glucose in blood. It used this glucose and stimulated synthesis of glycogen. Decreased gluconeogenesis in liver. Gluconeogenesis or synthesis of glucose, increased glycolysis in liver and adipose tissue, decreased the breakdown amino acids in liver, increased amino acids uptake and protein synthesis in muscle, liver, and adipose tissue, decreased lipolysis and increased lipogenesis and esterification of fatty acid in liver and adipose tissue. In this picture, we also see metabolism effect of metabolism by effect of carbohydrate metabolism, effect of lipid metabolism, and protein metabolism. And you see that insulin have anabolic effect, stimulated synthesis of protein, stimulated lipogenesis and stop lipolysis, and decrease glucose in blood metabolism. Insulin is special hormone which also insulin decrease glucose in blood, and it's used glucose for synthesis of stimulated synthesis of glycogen and increase utilization of glucose, or it's glycolysis, stimulated glycolysis. In this picture, we describe the deficiency of insulin and the central role of insulin in carbohydrate, lipid, and protein metabolism we can show by insulin deficiency in human. The cardinal manifestation of diabetes mellitus is hyperglycemia, which results from decreased entry of glucose into cells. Next, decreased utilization of glucose by various tissue and increased production of glucose by the liver, or it's called gluconeogenesis. Polyuria, polydipsia, and weight loss in spite of the adequate calories intake are the major symptoms of insulin deficiency. The plasma glucose level rarely exceeds 120 milligram in normal human. But when level of glucose will be increased till 180 milligram, occur glucosuria. Glucosuria, it is excrete glucose in the urine. The urine volume increased owing to osmotic diuresis and coincident obligatory water loss. It's called polyuria. And this is turn leads to dehydration of body, increased thirst. And excessive drinking, it's called polydipsia. Glucosuria causes a substantial loss of calories. The loss this associated with loss of muscle and adipose tissue result in severe weight loss in the spite of increased appetite, and it is called polyphagia. Polyuria, polyphagia, polydipsia, it is main symptoms of diabetes mellitus. Protein synthesis also decrease in the absence of insulin, partly because the transport of amino acids into the muscle reduced. The amino acids serve as a glucogenic substrates. Those insulin deficient persons are in negative nitrogen balance. Next, about lipid metabolism, the anti-lipolytic action of insulin is lost. But it is this part show lipid metabolism as is its lipogenic effect has plasma fatty acids level rise when the capacity of the liver to oxidize fatty acids to CO2 is exceeded by hydroxybutyric acid and acetacetic acid accumulated. It is ketone body production increased and form hyperketonemia and form ketonuria, excrete ketone bodies with urine. Then the organism initially compensates for accumulation of the organic acids by increasing respiratory losses of CO2, but if unchecked by the administration of insulin, severe metabolic acidosis supervenes and the patients dies of diabetic coma. Next hormone of pancreas, it is glucagon. Glucagon is a single chain polypeptide and consisting of 29 amino acids. Its hormone have polypeptide nature and circulated in plasma in the free form, since it does not associate it with a transport protein. Glucagon is inactivated by the liver, which has an enzyme that removes the first two amino acids from the N-terminal end, and also the secretion of glucagon is inhibited by glucose, an action that emphasizes the opposing metabolic role of glucagon and insulin. In general, the action of glucagon opposite those of insulin. Whereas insulin promotes energy storage by stimulating glycogenesis or synthesis of glycogen, insulin decrease glucose in the blood and used for synthesis of glycogen, and also stimulated lipogenesis and protein synthesis. Glucagon causes the rapid mobilization of potential energy of energy sources into the glucose by stimulating glycogenolysis or degradation of glucose, and that's why increased glucose in blood. And into fatty acids by stimulating lipolysis. Glucagon is also the most potent glucogenic hormone, and it is ketogenic. The liver is the primary target of glucagon action. Glucagon binds to the specific receptors in the hepatic cells plasma membrane, and this activated adenylyl cyclase, the cycle adenosine monophosphate generated activated phosphorylase, which increased the rate glycogen degradation while inhibiting glucogen synthase and this glycogen formation. There is hormone and tissue specificity in this effect, since glucagon has no effect on glycogenolysis in the muscle, whereas epinephrine is active in both muscle and liver. The net action of glucagon in the liver is increased glucose production, since much of this glucose exists in the liver. The plasma glucose concentration increased the liver plasma glucose concentration increases in the response to glucagon. Glucagon is a potent lipolytic agent. It increases adipose cells cycle adenosine monophosphate levels, and this activates the hormone sensitive lipase. The increased fatty acids can be metabolized for energy formation or converted to the ketone bodies, acetoacetate and hydroxybutyrate. This is an important aspect of metabolism in the diabetic, since glucagon levels are always increased in insulin deficiency. Now we talk about hormones of adrenal medulla, and the main hormone of adrenal medulla are adrenaline, noradrenaline, and dopamine. Its general names of this hormone catecholamines. The catecholamines, its hormone dopamine, adrenaline, or epinephrine and norepinephrine, and these all catecholamines derivatives of amino acids tyrosine. These amines are synthesized in the chromaffin cells of the adrenal medulla. Collection of these cells are found in the heart, liver, kidney, gonads, and central nervous system. The major product of the adrenal medulla is the adrenaline, or other name is epinephrine. Adrenaline and noradrenaline may be produced and stored in the different cells in the adrenal medulla and other chromaffin tissues. In this picture, we see the conversion of tyrosine to adrenaline. This process requires 4 sequential steps. First, conversion amino acid tyrosine to dopamine. Tyrosine is precursor of catecholamines, and tyrosine hydroxylase in the rate-limiting enzyme in catecholamine biosynthesis. Its first enzyme, tyrosine hydroxylase, which catalyzes this reaction, convert to tyrosine to dopamine, L-DOPA. In contrast to tyrosine hydroxylase, which is found only in tissue that synthesize catecholamines, next enzyme, decarboxylase, is found in all tissue. In the next, dopamine hydroxylase is a mixed function oxidase that catalyzes the conversion of dopamine to noradrenaline. And this enzyme have coenzyme. Here I not say about coenzyme pyridoxal phosphate or vitamin B6, and dopamine hydroxylase have used in this reaction ascorbat as an electron donors, and Cooper, which have in the active site. Next enzyme which catalyzed and reaction, it is enzyme phenyl ethanolamine and methyl transferases to form epinephrine to the epinephrine in the epinephrine forming cells of the adrenal medulla. This process occurs in the cytoplasm, and donor of these methyl groups will be amino acid tyrosine. Catecholamines circulate in plasma in a loose association with albumin. And the catecholamine act through two major classes of receptors. There are, next slide, there are distinct alpha adrenergic and beta adrenergic, and epinephrine bind to and activates both alpha and beta receptors, but noradrenaline at physiologic concentration primarily binds only to alpha receptors. Now, how adrenaline show his biology activity, mechanism action of adrenaline and effect of metabolism. Adrenergic receptors are coupled to the adenylyl cyclase systems. Activation which activate of phosphoproteins by cyclic adenomonophosphate dependent protein kinase and account for many of the biochemical effect of epinephrine in muscle and in the liver. Epinephrine stimulates glucogenolysis. Glucogenolysis is degradation of glycogen by activating a protein kinase that in turn activated the phosphorylase cascade. And phosphorylase enzyme, it's an enzyme which stimulated synthesis of glycogen. Conversely, phosphorylation of glycogen synthase decreases glycogen synthesis. In heart muscle, epinephrine increases cardiac output by increasing the force and rate of contraction, which are also related to increased cyclic adenosine monophosphate. In adipose tissue, epinephrine increased adenosine monophosphate, which converts the hormone-sensitive lipase into the active or phosphorylating form. This enzyme promotes lipolysis and release of fatty acid in the circulation, and these fatty acids serve as energy source in muscle and can be activate gluconeogenesis in liver and increased amount glucose in blood. In this table, you also see comparison of mechanism action of three main hormones. Epinephrine rapidly provides fatty acids as a primary fuel for muscle action. Next, mobilizes glucose as an energy sources for the brain by increased glycogenolysis and gluconeogenesis in the liver, by decreasing glucose uptake in the muscle and other organs, and also decreases insulin release, which also prevents glucose from being taken up by peripheral tissues and thus preserve it for the central nervous system. Now we describe other part of adrenal gland. It is adrenal cortex. The adrenal cortex synthesizes dozens of different steroid molecules, but only a few of these have biologic activity. These sort into three classes of hormones: glucocorticosteroids, mineralocorticosteroids, and androgens. These hormones initiate the action by combining with specific intracellular receptor and This complex binds to specific regions of DNA and regulate the gene expressions. This results in altered rates of synthesis, a small number of protein, which in turn affect a variety of metabolic processes. Gluconeogenesis in glucocorticosteroids and natrium and kalium balance. The hormones of the adrenal cortex, particularly the glucocorticosteroids, are an essential component of adaptation to severe stress. The mineral corticosteroids are required for normal natrium and kalium balance. Synthetic analyses of both classes are used in the clinic. The glucocorticosteroids are 21 carbon steroids with many actions, the most important of which is to promote gluconeogenesis. Cortisol is the predominant glucocorticosteroid in human, and it is made in the zona facilate. Corticosterone made in the zone of facilate and mineral corticosteroid are also 21 carbon steroids. The primary action of this hormone is to promote retention of natrium and excretion of kalium and hydrogen, particularly in the kidney. The androgen procures dehydroepiandrosterone. It is next androgen, hydroepiandrosterone. These steroids are converted into androgens in the extra-adrenal tissue and become pathologic sources of androgens when specific steroidogenic enzymes are deficient. Estrogens are not made in the normal adrenal in significant amounts, but in certain cancer of the adrenal, they may be produced. And the androgens of adrenal origin are important precursors of estrogen in the post-menopausal woman. The adrenal steroids, all adrenal cortex hormone have steroid structure, and all hormones are synthesized from cholesterol. All mammalian steroid hormones are formed from cholesterol via pregnenolone through a series of reactions that occur in the mitochondria of endoplasmatic reticulum of the adrenal cells. Now describe the biochemistry effects of corticosteroids. Glucocorticosteroids hormones are named for their ability to promote glucose production. And glucocorticosteroids promote hepatic glucose production by increasing the rate of gluconeogenesis by releasing amino acids and gluconeogenic substrate from peripheral tissue such as muscle and lymphatic cells through catabolic action. Glucocorticosteroid hormones also inhibit glucose uptake and utilization by extra hepatic tissue, so that the net effect is an elevated plasma glucose level. Also, this hormone enhances gluconeogenesis by increase the amount of several K hepatic enzymes. The induction of several enzymes that catalyze rate limited step in amino acid degradation. Glucocorticosteroid increase glucogen deposition in the liver. About protein metabolism, glucocorticosteroids generally have anabolic effect on protein and RNA metabolism in liver, and catabolic effect at other sites, including muscle, lymphatic tissue, adipose tissue, skin and bone. Now about fatty metabolism or lipid metabolism, they excessive amount of glucocorticosteroid promote lipolysis in the some areas of the body and lipogenesis in face, other area. Plasma-free fatty acids levels increase in humans given glucocorticosteroids. The net increases of plasma-free fatty acid coupled with the enchant conversion to ketones and formed of ketosis. Mineralocorticoid hormones act in the kidney to stimulate active natrium transport by distal convolute tubulus and collecting tubulus. The net result being natrium retention. These hormones also promote the secretion of kalium, hydrogeniols, and ammonia by the kidney and affect ion transport in other epithelial tissue, including sweat glands, intestinal mucosa, and salivary gland. The secretion of cortisol is dependent of adrenocorticotropic hormone, which in turn is regulated by corticotropin-releasing hormone. These hormones are linked by a classic negative feedback loops. The general features of aldosterone action are similar to those of other steroid hormones. Target cell contains specific receptors that bind aldosterone. Receptors that bind aldosterone with high affinity and are found in the cytoplasm and nuclei of target cells. Corticosteroids are avidly bound to corticosteroid bindings globulins, the plasma, glucocorticoid transport proteins, while aldosterone has no, aldosterone has no specific carrier proteins. Disorders involving glucocorticoid hormones. Primary adrenal influence Addison disease results in hypoglycemia, extreme sensitivity to insulin, intolerance to stress, anorexia, weight loss, and severe weakness. Patients with Addison disease have low blood pressure and decreased ability to excrete a water load. They often have plasma natrium levels are low, kalium level are high, and blood lymphocyte and eosinophil count are increased. Such Patients often show increased pigmentation of skin by the compensatory secretion of adrenocorticotropic hormone. Secondary adrenal insufficiency in due to a deficiency of adrenocorticotropic hormone resulting from tumor, infarction, or infection. This results in a similar metabolic syndrome without hyperpigmentation. Glucocorticosteroid excess, commonly called Cushing syndrome, is usually due to the pharmacologic use of steroids, but it may result from adrenocorticotrope secreting pituitary adenoma. This patient have hyperglycemia or glucose intolerance, or both of them, because of accelerated gluconeogenesis. Related to this are several protein catabolic effects, which result in thinning of skin, osteoporosis, extensive lymphoid tissue involution, and general nitrogen balance. Thank you for attention.